Hello again, everyone, and welcome to the Science of Childhood Cancer lecture series hosted by the St Jude Comprehensive Cancer Centre. My name is Charles Mulligan. I'm in the Department of Pathology at St Jude. I'm also co-leader of the Haematologic Malignancies Program and Deputy Director of the Cancer Centre. Before we get going, I just point out that this is now the midpoint, the 8th week of a 15-week series, a new series for St Jude, which we developed in a time of COVID as an experiment to foster scientific engagement and interaction in a virtual space, really specifically devoted to various aspects ranging right through to fundamental biology to survivorship in pediatric cancer. This has been really terrific, we think. We've had over 3,200 registrants as of this week and attendees from not just North America, but all over the world. And it's my pleasure today to welcome Dr. Yannis Ifantis from New York University Grossman School of Medicine. Before I introduce him, just a couple of housekeeping points. There will be moderated questions at the end of the talk. So if you have questions at any time during the talk, please use the chat function in WebEx and send questions to all panelists. Or if needed, you can email them to socc@stjude.org. If you have any technical issues during the talk, please contact SOCC support. That's one word, SOCC support @stjude.org. Yannis will leave time for questions before the top of the hour and has kindly agreed to stay on for about an additional 30 minutes after the close of the official event, if anyone would like to stay for additional moderated discussions. So please remember that. So now it's my pleasure to move to the main event to introduce Dr. Yannis Ifantis. He is Professor and Chair of the Department of Pathology at the NYU School of Medicine. Originally from Greece, Yannis undertook graduate training at the Necker Institute in Paris and postdoctoral training at the Dana-Farber Cancer Institute, including training with the legendary immunologist Harold von Boehmer. He initially joined the faculty of the University of Chicago after his training and then has been at NYU now for, I think, about 14 years. He has a number of and diverse interests that are directly relevant to pediatric cancer, particularly lymphoid and myeloid leukemia. These include the regulation of hematopoiesis, epigenetic mutations and regulation, the role of non-coding RNAs in tumorigenesis, the nature and composition and regulation of the bone marrow microenvironment, and the topic of today's talk, 3D chromatin structure and organisation. So again, Yannis, welcome, and thank you for joining our seminar series. Over to you.
Thank you so much, Charles. It's a pleasure to be here, and I mean, I know that it's an online event, but I think the exciting part of the online event is that we have 100 plus participants, something that we could never done before at a normal setting. So it's just a silver lining, I guess. Let me just share my screen. Okay, so as Charles mentioned, I would like to tell you some of our recent work on 3D chromosomal landscapes in T-cell leukemia. And I'm sure most of you know that our chromosomes are much more complicated and twisted that we actually thought initially. And there's basically this idea that you have to pack almost 2 meters of DNA basically in a small cell. So the way to do that is to actually have a 3D structure, and the nucleus is basically organized in different chromosome territories. The territories are organized in compartments, as I'm going to show you in a couple of slides. And the compartments are by themselves comprised by what we call topological associated domains, which also include hundreds or even thousands of individual loops. Around these loops, you can have proteins, structural proteins like cohesin and CTCF. So what is a TAD? And I'm mentioning TADs because we're going to discuss a little bit more about them. It's basically an area of higher probability and frequency of interactions on a chromosome. Imagine a neighborhood of genes and enhancers that they basically interacts with each other. And what you actually have is that you do have an insulation and you have boundaries between different TADs. And this is where exactly on these boundaries we do find things like CTCF and cohesin, as I mentioned before. So you do have higher TADs, and sometimes you even have smaller structures that we actually call sub-TADs. And computationally, the way that we visualize them are basically by these triangles, in this case, red triangles, as you can see in the screen. How do we generate this computational information? We do it by basically using different types of chromatin conformation capture assays. And today I'm going to mention 3 of them. I will mainly talk about Hi-C, which is every interaction with every interaction. And I will validate findings using 4C, which is basically one area with other areas of interaction. And we're going to use a little bit of Hi-CHIP, which is basically something like a Hi-C, but before the step of pull down and ligation, you basically have a step in which you use an antibody. And in our case, for example, we're going to use an antibody that recognizes K27 acetylation that looks for active areas on chromosomes. These techniques were introduced by people like Lieberman Eiden and his beautiful work during the last decade or something, and several others in the field pioneering newer techniques and newer ways of analysis. So as Charles mentioned, one of our first labs in the area of hematopoiesis was basically lymphopoiesis. So basically, this idea of programs of differentiation that start from a stem cell in the bone marrow progress through a multiple potential progenitor stage, and eventually they will generate all the immune system, and specifically the lymphoid part of the immune system. So T cells, B cells, and K cells. As most of you probably know, different types of leukemia can be assigned to different differentiation stages. And today we're going to focus mainly on T-cell acute lymphoblastic leukemia, so TLL, which it's a disease of T-cell progenitors that comes kind of like later in differentiation. We're going to mention ETPLL, which, as the name says, early thymocyte progenitor. It is initiated somewhere between a stem cell, a multipotential progenitor, and an early T-cell progenitor. And of course, different stages, different types of acute myeloid leukemia can also come from similar early stages of differentiation. And we'll mention that later, but ETPLL is a disease that has been introduced and worked out at St. Jude's during the last decade plus by people like Dario Campana, Charles Mulligan, and Brian Sorrentino, and others. And I will refer to some of their papers. TLL, or conventional TLL, is basically a disease that comes from transformation of T-cell progenitors, thymocytes most likely, and it is characterized by the activation of Notch signaling. This is actually the first type of leukemia that we studied in my own lab, more or less when we started. That's 16 years now, and it is characterized by Notch mutations and Notch activation. So that's why at some point in my talk, I will refer to Notch inhibitors, which are basically drugs that block cleavage and activation of the pathway. And during the years, my lab and several other labs have actually characterized the pathway at a lot of different stages, from downstream transcriptional targets, and I'm going to mention several of them, including MYC, which is a canonical target of Notch in T-cell leukemia, to regulation by the ubiquitin proteasome pathway, and we have worked a lot of EPXW7 as an E3 ligase, and several other elements of this pathway. So the first thing that got us into topology of chromosomes is basically work that we have done almost 8-9 years now, with Thomas Trimarchi in the lab and Panos Gianchristos and Aris Tsirigos, where we have actually characterized the IGF-1R locus and a long non-coding RNA that it is now called LUNAR1, and we have seen that it's actually part of a TAD that basically sits on this area and enhances interactions within this chromosomal domain. I mean, we were not the only ones. Several papers after us in TLL have basically characterized 3D events or transcriptional epigenetic events that are important in this particular disease. And of course, work from the Mulligan lab has characterized mutations on epigenetic regulators, or more specifically, regulators like CDCF, which basically set the boundaries, not only for TADs, but for individual looping events. So related to that, what my lab has done in the past was basically to generate animal models of loss of cohesin genes. And we have shown that loss of cohesin can actually be important for transformation. It can act as a tumor suppressor, and it can lead to myeloid neoplasms. And we are not the only ones. Our friends and collaborators in Ross Levine's lab, together with us, described very, very similar findings. So basically what I'm going to tell you today, we're going to address the following questions. Is cellular transformation accompanied by 3D chromosomal changes? Is drug treatment able to reverse aberrant 3D chromosomal organization? And at the end of the day, what is the role of 3D chromosomal organization for the differentiation and self-renewal of normal, but also leukemia stem cells? So the team behind this work was Raj and Andreas, two of our postdocs, one a biologist and the other computational, and of course Aris Tsirigos that has worked with us. We have worked with him for the last decade, and we are using his own computational methods to analyze all of this data. So the setup of the study was very simple. We have taken normal peripheral T cells, we have taken peripheral TLL cells, and we've also taken thymocytes. And we have done all of these assays that I mentioned here, plus now we have data on high chip with K27 acetylation. Basically, the analysis was done using Aris's high-C bench, which is a beautiful method of analysis, but also visualization of 3D landscape data. The way that we do this analysis is the following. We're looking for compartment changes. We're looking for stat structures themselves. We're looking to call individual loops. And more recently, and that's also work that Aris has done, is we're actually looking at what we call enhancer hubs on chromosomes. So just a few words, because I'm not going to stay with that, on compartment changes. Basically, people have found that parts of the chromosomes can be found in different compartments. So basically, some compartments can be nuclear lamina, close to nuclear lamina, and these are compartments that are more heterochromatinized and silent, and other compartments which are more in the center of the nucleus are much more transcriptionally active compartments. So what happens from our own study is that we have seen the same thing. We have wide compartment changes when we compare normal T cells with TLL, and we have A to B, and by A is active to inactive, and we also have B to A compartments changes. And what is actually more interesting is that go compartment by compartment. The one thing that I can tell you is that as we expected, A to B basically led to loss of expression, and it was even more prominent that B to A compartment changes led to clear gains of expression of genes within this same compartment. So if we get out of compartments and now look at individual TADs and what we call insulation scores, so how strong the TAD scores and the interactions are within TADs, we had a surprise. And the surprise that we had that when we started sequencing, I mean, if you look at this PC plot here, we put in two different cell types. One is a T cell, normal, and the other is a leukemia. But we got out three different clusters. So for a little while, we were basically questioning this data, and we're trying to understand why that actually happened. And then, of course, we remembered data that came from Charles's lab and Adolfo's, Ferrando's lab, and several other labs, suggesting that indeed, you know, TLL is not one type of disease, but it's at least two wide type of disease. So very easily, when we went back to our gene expression data, we could see that basically the PC plot that we got on insulation score from high C was able to diagnose between a conventional TLL, ETPLL, and T cells. And these are studies that we're doing now. So this is kind of an updated slide with unpublished data on where are we standing at this point. And you can see that now it's a little bit more complicated. We have the T cells on one side. We have double positive thymocytes somewhere in the middle. We have CD34s somewhere here. And then we have a neighborhood of ETPLLs, something that you know, pathologists might call near ETPLLs, and then kind of more conventional TLL samples. So very interesting, because just high C, just insulation scores could basically identify different subtypes of TLL. And now we've actually expanded this study. I don't have it in this slide, but you know, if you add BLL in this slide or AML in this slide, they basically are separated very nicely in different areas of our PC plots. I don't probably have to tell you in this particular audience what ETPLL is. It's a smaller subtype of TLL, probably 15 to 10 to 20%. And work from St. Jude's and other institutions in the past suggested that it is a more high risk of subset of disease. And what we know for conventional TLL does not particularly apply for ETPLL at a lot of different levels. You know, ETPLL is characterized by some mutations which are more frequent than TLL. It doesn't have that many Notch mutations or PXW7 mutations. And even the cell of origin is different. And it was shown before by some of your work that it is actually closer to a stem cell than actually a T cell progenitor. So if you actually now use high C to try to predict cell of origin, and this is, you know, some initial published data that you see, we can see that basically conventional TLL it's closer to a T cell progenitor than it is to double positive cells or peripheral T cells. And this is the way that we compare some of these studies. We compare what we call intra-TAD activity. So basically how strong interactions are within the TAD. And today I'm going to mainly talk to you about T cells and T cell leukemia, but we have done a lot of analysis between T-cell leukemia and double positive cells, T-cell leukemia, conventional and ETP-LL, and all type of combinations in this study. So basically what we call changes in intra-TAD activity, you can have, for example, a red hot TAD with a lot of interactions in one cell type, and then in the other cell type, this TAD could become a little bit less hot. So for example, you have less interactions within the TAD, but you could have the opposite. So basically, if you just compare peripheral T cells and transformed TLL cells, you can see that you have gain of TAD activity and loss of TAD activity. And some of very interesting genes can be found on the gains of TAD activity. And a lot of these genes are basically canonical Notch1 targets, as we've expected. So Icarus genes, MYC itself, ABCDD1, et cetera, HESH1, Deltex, all of these genes can be actually found in this particular category. Interestingly, as you would expect, intra-TAD activity correlates very well with differential RNA expression. So gain of intra-TAD activity and looping is correlating with increased expression of these particular genes, and I will show you a few examples. The other thing that is really interesting is that intra-TAD activity gains correlates with stronger CTCF binding. So you get higher CTCF binding peaks around it, or better insulation scores, just because now the TAD activity inside, it's much more stronger, and when compared to basically looping events that lead to adjacent TADs. So let me show you a couple of examples of gains of TAD activity. It was shown before, and I think this is one of your papers, that in ETP-LL you have several regulators of the phenotype of the cells, and one of them is FLT3, which is transcriptionally upregulated in ETP-LL. And if you now go to high-C data, it looks a little bit like that. So look at the gained basically activity of this particular TAD. Here we can actually quantify the sum of TAD activity, and that correlates very well with increased RNA expression of FLT3 in ETP-LL. On the flip side, if you look at conventional T-cell leukemia, we can basically look at several Notch genes. I chose to show you one of them here that's called ABCDD1. Canonical Notch target. You can see here basically the gain of K27 acetylation of this particular locus. And look at here, primary TLL samples and a TLL cell line, very strong TAD and high TAD activity, and correlates very well with increased expression of RNA in this RNA sequencing experiment. And this is also log2 change in this particular scale. So the other thing that we have looked at, it's what Aris and his people called TAD boundary changes. So you can have different combinations. So you can imagine that you can have two smaller TADs, which are separated in, let's say, your normal cells. And in your leukemic cells, now you can have a gain of a larger TAD, so what we call a fusion event. And you can have the opposite. You can have a larger TAD that actually breaks into small separated TADs. And I'm just going to give you today one example. And this is the good old MYC locus. I've told you before that MYC, Adolfo, Ferrando, and John Astor and several others have actually shown that it's a canonical Notch target. So let's look at the locus. So first of all, before we go into the MYC locus, several colleagues during the years, including Adolfo Ferrando, Francois Spitz, Andreas Trump, has actually characterized enhancer elements that are downstream on the MYC coding region itself. And some of them are particularly active, not only in stem cells in hematopoiesis, but also in T-cell leukemia. And Adolfo has done beautiful work characterizing an enhancer that he called NME. So this is the locus. Basically, bear with me, complicated slide. This is the locus. This is NME here. And you see the K27 acetylation and the attack sequencing reads. This is the second enhancer element or super enhancer element with the attack sequencing reads. And now, if you look at the actual TAD structure, it looks a little bit like that. So normal T cells, human T cells, have actually two smaller TADs. This is where MYC is. This is where the super enhancer cluster is. So basically, in normal T cells, there's very little interaction between these two sides. If you look at T-cell leukemia, it looks a little bit like that. Let's look at the Jurkat cell line. You now have a fusion, and you do have a large TAD, and now you have very strong communication between MYC itself and the super enhancer elements, which is, this is the gain of interactions that you can see here in red, and this is translated in a gain of RNA sequencing reads and RNA expression. So how is that happening? How do you have a fusion event on a TAD? Let's focus on the locus here, and let's look at this pink line. If you look at normal T cells, you have this very high, strong CTCF peak, which is, we believe, what separates the two smaller TADs. What happens in TLL cells, and you can see it here, 1, 2, 3, in a cell line, is basically that this CTCF binding site or CTCF binding event basically just goes away. And by going away, you don't have the insulation, you don't have the boundary, and you have the fusion events, the event that leads to this higher expression of MYC. We can prove that several ways. The first thing that we've done is basically we've actually visualized by using FISH, and you can see that in normal T cells, look at the two different dots here. The promoter is far away from the enhancer cluster. If you look at T cell leukemia cells, they are basically on the top of each other. They are fused, they're on the top of each other, they interact, and that leads to higher expression of MYC in TLL cells. We can validate that different ways. The other way that we have done that is by 4C. And most of you probably are not used to 4C reads, so bear with me. This line is normal T cells. This is exactly where the CTCF side was. And basically what we see is that this is the first small TAD. It starts upstream of MYC. You see the interactions with PVT1 in normal T cells, which is a non-coding RNA, and they continue a little bit further down. And all interactions are basically blocked where the CTCF site and the insulation boundary is. And you don't see, you see very little leakiness of interaction with the actual enhancer. If now you look at TLL cells, you can obviously see fewer interactions upstream here and a lot of interactions that are situated on the top of both enhancers elements that I've talked about you before. So this is how it looks in real life, not in a cell line, but in patient cells. So again, the same thing. So normal T cells, they have this huge boundary here and the interactions stop, as you can see here. And if you look at TLL patient samples, they have very high interactions, specifically between the promoter of MYC and the super enhancer elements that are located in this area. So the second question that I want to address today is, can a drug basically fix all of these things? Okay, I mean, and I chose a drug that is not in a clinic at this point, but is a pathway targeting agent, and this is gamma secretase inhibitors. Gamma secretase inhibitors have their own story in TLL. That we can discuss if you want later. The one thing that I want you to keep in mind is that it's a very efficient inhibitor of the Notch pathway. So it blocks cleavage, Notch intracellular doesn't get into the nucleus, doesn't bind to the targets, doesn't bind on MYC, etc., etc. So we decided to use gamma secretase inhibitors, and when we've done that, it looks a little bit like that. So we have seen that, as we expected, we see loss of enhancer activity and loss of K27 acetylation and K4 monomethylation, specifically on enhancers that are controlled by Notch1. And this is what Warren Pear and John Astor have called before Notch dynamic sites on the chromosomes. And not only you lose K27 and you lose activity of the enhancers, but you actually lose interaction. So if you look at classic Notch target, like I told you about the IGF-1R, lunar run locus, or the APCCD1 that I described before, these are the ones that are basically down here. And they're not only they lose K27 and activity of enhancers, but they lose basically looping between enhancers and promoters. A couple of more examples. IGF-1R here, look at the enhancer. This is the intronic enhancer that we have characterized before. And look at the drop in the enhancer, black line, green line, and the net loss that you have in this particular site. APCCD1 that I've told you before is another one. Look again, the enhancer element and the loss specifically on this regulatory side. However, one big surprise that we had was that not all loci are equal when it comes to inhibition. So if you look at our favorite MYC locus and you add a gamma secretase inhibitor, you lose a little bit, but not much of your K27 from the super enhancer cluster. You do lose binding, not totally, but you do lose binding of Notch1. So this is indeed a dynamic site, but for some reason, and expression is decreased, as you can see in this gross sequencing experiment. But basically interaction and looping does not particularly change, as you can see here. So this is what we've seen with K27 acetylation. If you look at the enhancer area, there is a decrease, but it's not spectacular. It's not as strong as some of the examples that I've shown you before. And MYC is not the only one. I mean, we have seen it in other loci. And another example is basically Helios, one of the Icarus factors, which is one of the classic Notch targets. And again, K27 is not decreased. Notch is going away, not totally. And interactions basically between promoter and an enhancer, they stay at the same level. So why is that? Why do we have enhancer elements and elements on DNA that are immune from drug treatment? So to address that, we've done several things. The one thing that we've actually tried to do is that we try to see and predict what binds there. What binds on these areas that are sensitive and how that compares with the insensitive ones and vice versa. And as you can see here, we looked at different things. P300, mediator, BRD4, the cofactor of Notch, RBPJ, RANKS1, ETS factors, you name it. One thing that was particularly interesting was binding of a kinase that's called CDK7. And as you can see here, CDK7 was very strongly depleted, specifically on enhancers that are basically insensitive for NOTCH1. And I mean, these enhancers were MYC and Helios and several other genes, but today I'm just going to focus on this second part and show you MYC and Helios only. So what is CDK7? CDK7 is one of these transcriptional CDKs. Its job is to basically phosphorylate the CDD of RNA pol 2 at serine 5, and basically is very important for initiation and elongation steps during transcription. And what is really interesting is that CDK7 has been proposed before that is important for the growth of T-cell leukemia cells. This is the work that came from Nathaniel Gray in his lab a few years ago. And the Gray lab actually has introduced an inhibitor, a very potent and specific inhibitor for CDK7, which is called THZ1. And as you can, as you can actually see here, you get pretty nice responses both for TLL and ETPLL cell lines, and they have also used it in vivo with different cell line transplantation models. So what we've done is that we used THZ1, and what we have seen is that when we add it to T-cell leukemia lines, several things happen, but in general, we have a decrease of K27 acetylation from enhancers in general. This increase of K27 acetylation goes together with loss of looping and interactions in these particular areas. And of course, the big question is, so what happened to canonical, what happens to canonical targets of NOTCH, and especially targets that are insensitive to NOTCH inhibition itself? So let's look at them. This is MYC on Jurkatz, which is a kind of classical TLL line. Control and THZ1 treated, and you can obviously appreciate that the whole super enhancer containing area is the interactions are basically significantly decreased in response to CDK7 inhibition. We have done it with several cell lines and primary cells. This is another cell line that is characterized by the Ferrando lab that's called CATL1, and you can see exactly the same thing, loss of interaction on this particular area. And we can actually visualize and measure the interactions by doing FISH analysis. So basically, this is untreated, and you can see here, as I've shown you before, the two probes are kind of on the top of each other, and you can see the separation that we actually get when we add the CDK7 inhibitor. And this actually happens in additional gamma secretase insensitive enhancer areas, not only on MYC, and I'm showing you here Helios. Look at the enhancer again, and look at the drop of the expression, of interaction that you see in this particular area. So basically what I've shown you, the paper came out a couple of months ago. The paper came out in the spring. And basically what we claim with some of this data that I've shown you is that first, cellular transformation, as we expected, is accompanied by changes in 3D chromosomal landscapes. That 3D organization changes, we believe that is a novel way to alter activity of oncogenes and tumor suppressors, and I've shown you a couple of examples with MYC, for example. that drug treatment can correct quote-unquote oncogenic or aberrant 3D organization. But we believe drug combinations can be more efficient in basically achieving at reversing some of these effects of oncogenesis. And there is an emerging notion of 3D regulation of oncogene and tumor suppressor activity in cancer in general. And these are studies that not only coming out from our work, but there's several other labs in different types of cancer and transformation that have published recently similar data. So it's, I mean, for us, it's very exciting. It's obviously kind of a brave new world. And we're excited to see what kind of lies ahead. I can take maybe 2-3 minutes and tell you about things that we have in mind and things that we're doing in the lab currently. So one of the things that we're particularly interested is we are trying to predict differentiation transitions based on 3C data, so high C and high CHIP data. Of course, cell numbers sometimes, complicate our efforts, but there's this fine differentiation transitions between stem, multipotential progenitors, committed progenitors, and terminally differentiated cells that we believe must basically translate in changes in 3C data and 3D interactions, and we're trying to go for it. And I just to give you one example, and that's because this is what I call a St. Jude based example, and this is the switch between CMYC in conventional TLL and NMYC in ETPLL, and this is work that Brian Sorrentino and Charles Mulligan have done in the last few years, and basically what they have shown is this, as I said, switch from a CMYC-based conventional TLL to a more NMYC expressing ETP ALL. So now if we look at CMYC, and you know this locus because I've shown you this locus before, and you do high C in hematopoietic stem cells, multipotential progenitors, commonly for progenitors up to or down to double positive cells or single positive cells, you see this very interesting. I mean, MYC is in general an area of a lot of interactions, but you can see that hematopoietic stem cells that are more quiescent, they have less of these interactions. CLP is the same thing, multipotential the same thing, and there's more basically expanding progenitors within the thymus that have higher degree of interactions, and maybe this is why we see TLL having this very intense interactions in this particular locus. So what happens now if you look at ETPLL, okay, and if you look at the NMYC locus? This is kind of the opposite. So NMYC locus is very interesting. My lab has recently characterized an enhancer element in the NMYC locus that we call NMRE, and we have some work on this particular element. But if you consider the TAD basically that contains MYC and its super enhancer, and you look at strength of interactions and you correlate it with MYC gene expression, I mean, look at that. The highest interactions are, of course, are in ETP cells themselves, stem cells, early progenitors, and basically drops as the differentiation progresses, and you switch from NMYC to CMYC. So this is just one interesting example that can connect with the work that some of you are doing or you are interested on, but there's several other examples during T-cell development that are obviously, really exciting for us, and we follow them. And we're not the only ones. Ellen Rothenberg and Tom Tagon and others have done similar work in different loci in the last few years. So basically, I'm gonna stop here. This is the reference to one of the papers by Brian and Charles on NMIC and ETPLL. And I will stop here and basically say that most of this work, or actually all of this work, was done, actually started with Panos Zachristos, who is now at Northwestern. And it was completed and finalized by Raja and Andreas. It was done basically under the supervision of computational analysis of Aris Tsirigos, several people from my own lab, and we could not do it if we didn't have the help of several people, Adolfo, Elizabeth, Peter, and Jane Skok and Tim Leonet here at NYU. So I'm gonna end here, stop sharing, and get questions. So thank you so much.
Thank you, Yannis, for a terrific talk. That was really absolutely fascinating to see how this field is moving and moving so quickly. We have plenty of time for questions. I think almost 12 minutes now before the top of the hour, so we can launch into them. The first question is from Janice Ribity, which is there a model where longitudinal analysis can be exploited to demonstrate the progression of the TAD fusion or fusions?
I'm trying to find the question on the chat because I wanted to ask what do you mean in this case by longitudinal? You mean during differentiation? Let me see. Is a model? Yeah, I mean, the process of TAD fusion is Janice is really interesting and exciting. I mean, for us, the question here was, how do you get actually this TAD fusion? And to be quite frank, we haven't really addressed that totally. So I show you some data in which you have this kind of progressive loss of CDCF binding in this particular area. We have done a lot of work to try and understand how that happens. So with Peter, if I'm being able, we have actually looked at DNA methylation changes in this particular locus. And we haven't actually seen any kinetic differences in the changes of DNA methylation that can actually explain it. The only difference in this particular area that goes together with the lack of the loss of CDCF binding is basically the appearance of open chromatin. So on ataxicon cystatis, we see opening of this particular element, suggesting binding of CDCF and insulation. But we haven't done any quote unquote longitudinal studies in this area.
Okay, so from Barthélemy Diouf, can 3D organization predict treatment efficacy, relapse, and outcome?
That's a great question. We are doing it currently. So we have already done high-C and high-CHIP on primary samples comparing diagnosis and relapse. I don't have a clear answer. I have loci that we're following up and they're obviously changing. Our expectation is that the answer is yes, but I don't have data to show you at this point.
Okay, this next question is somewhat related, I guess, and that is there any evidence of or investigation into clonal heterogeneity that can be characterized by variations in this 3D structural disruption? Or are those disruptions primarily correlated with the essential oncogenic drivers?
I mean, that is a beautiful question. I mean, so many people have done, including some of the people here, have done beautiful work on clonal hematopoiesis, clonal heterogeneity within leukemia. I expect that there are obvious 3D changes if you compare different clones, just because clones are characterizing by different chromosomal, changes or mutational landscapes. The limitation of the moment is single cell analysis. I mean, we can do single cell analysis at the RNA sequencing, and we're now doing a lot of attack sequencing in the lab, and I know that people are doing single cell methylation, but and there have been a couple of high-profile papers on single cell Hi-C, but to reach the point of clonal heterogeneity, you would need single cell Hi-C and correlation with genomic changes. We're not there yet, let's say. But my expectation would be that we're probably going to be there pretty soon. I have one person in my lab basically trying to look at single cell Hi-C and Hi-CIP, but we're not there yet at this point.
Okay, so the next question is from Yogesh Jungana, I think. I haven't mispronounced the name. The effect of drug treatment on nuclear mitochondrial gene organization. As in Notch mutant TLL has altered mitochondrial activity. Do they show similar switch between lamina-associated heterochromatin and euchromatin?
So my lab, I mean, that is a very cool question. So Yogesh, my lab is doing a lot of mitochondrial work, mainly when it comes to treatments with agents like venetoclax, for example. So we have tried to look at this particular question. Mitochondrial DNA is actually pretty short. So we have never succeeded at so far to actually get so fine resolution results that I can talk about differences on mitochondrial DNA. But I mean, that's obviously another frontier, and there's a lot of data on mitochondrial DNA conformation that came out like a few weeks ago. So we're gonna revisit that definitely.
Okay, so from Kun Q Yan, you mentioned using Hi-C to predict cell of origin. Should we take into account the population nature of Hi-C data and do some kind of deconvolution?
Yeah, absolutely right. We should definitely do that. But I think what we should actually do is two things. Either we can start with, let's say, fine flow sorting of very small populations of very highly purified stem and progenitor cells, or we should go down to single cell Hi-C to basically avoid looking at larger populations, which could actually kind of like muddy the waters of our analysis. So I absolutely agree with you that we have to do that.
Right, so we're still waiting for additional questions to come in, so please use the chat function if you have them. While we're waiting, Giannis, I'm going to ask a question or two if I can. Again, great talk. I really enjoyed it. So, the ETP question is dear to both of our hearts.
Yes.
And based on now quite old data from really, a decade ago when we first started looking at this, based on transcriptional profiling, although from the mouse it looked like ETP leukemia might have been reflective of the ETP stage of thymic development, comparison to human genomic data really suggested that it may be part of the spectrum of stem cell leukemias or, earlier stage of development than the ETP is the cell of origin. Have you got any thoughts about this, looking at this additional layer of data? If you go back to your schema of the stages of development, where do you think the cell of origin is?
I mean, look, ETP is called ETP because, as you said, of kind of historical reasons. We should probably, no, actually, we should not revisit the name, leave it where it is. But yeah, I obviously went to this type of analysis because I was aware of data that came out from your lab, but also from other labs that actually agreed with this idea that if you look at human differentiation, ETPs are closer to HSCs than a DN subset in the thymus. And I think I've shown some data. I don't know if you've seen. I'll be happy to share my screen if you want it, or just send you the slide. That if you add, because you remember initially we've done this very crude analysis by adding T cells and TLL cells, and then we had ETP cells and T cells and TLL cells. And so now, and this is unpublished, when we add double positive cells, thymocytes, and CD34s, it's obvious that ETPLL is much closer to HSC, so CD34s, let's say, because even the HSC, I believe that if it's an HSC or it's an NPP, it's still an open question, but it's much closer to an HSC and NPP stage than anything else. So the three data that we have so far absolutely agree with that. We haven't published it on anything, but it's obvious from our analysis.
Okay. What about CTCF mutations? You mentioned them as being recurrently mutated in TALL. What do you think is the functional consequence? Have you looked at that?
So we have collaborators that are looking at that. In vitro modeling so far by introducing mutations and basically looking at binding of DNA suggests that some CTCF mutations either abrogate binding or they change the recognition of the motifs. So we believe that this is actually what happens here. Most of the mutations, if I remember well from your own data, of CTCF and TLL are loss of function, a kind of like clear loss of function. So an experiment that we have with actually Ari Melnick at this point going on is that we have normal mouse TLL driven by NOTCH and CTCF heterozygotes and homozygote knockouts. And we basically try to see, do you get faster leukemia? Why do you get faster leukemia? And if that proves indeed that CTCF acts as a tumor suppressor, as all of the sequencing data suggest, then maybe we should look at more fine mutations. But from as I remember, in TLL, most of the mutations are loss of function.
They're in the zinc fingers and predicted to be loss of function. So yeah.
So that's why I think for us, when it comes to animal modeling, we started with loss of function, and then we can introduce mutations exogenously by lentiviruses or something like that.
Okay. We have a couple more questions, but we're at the top of the hour. So for those that are signing off now, again, I want to thank Yannis for his fantastic talk and officially close the event. But again, please stay if you want to participate in the ongoing discussion. Just one other shout out as well, the next talk. So the next one for Thursday, October 8th is from Paul Northcott of St. Jude on medulloblastomics 2020, where he'll, I'm sure, present a tour de force on his studies from thousands of patients. And October the 15th has Crystal Makel from Stanford speaking in the immunotherapy sub-series of the Science of Childhood Cancer. So again, thank you all for joining. Yannis, a question from Harris Lazarus. Nice talk, Yannis. Could you elaborate a bit on enhancer hubs and your approach to detect them?
So Harris did his PhD here with me and Aris, and he was part of our Nature Genetics work. So Harry, as you know, subsequently, Aris and collaborators from Cornell, they have done some beautiful work that was published this year at Nature Cell Biology, defining enhancer hubs. So basically areas on chromosomes, mainly enhancers, that don't have basically interaction with one site or one promoter, but that basically have interactions right and left with multiple sets of other elements and other genes. So we are particularly interested on them, and we have done work so far on ETPLL and conventional ALL. I mean, the only thing that I can say at this point, because the analysis is very early, if you want to see how the analysis is done, of course, you can talk to Aris, but you can go back to the paper that he has published a few months ago at Nature Cell Biology. I mean, a lot of the epigenetic regulators that characterize conventional TLL or ETPLL, or some of the signaling molecules that we know that are very active, downstream of IL-7 receptor and things like that, they score very high, particularly high in our enhancer hub analysis. It reminds us a little bit or very much the super enhancer analysis that Harris is actually now at MIT, that your MIT friends have actually done there. So, I mean, in one way, you know, if you call it an active enhancer or a super enhancer or an enhancer hub, you are looking at very similar things. But we are trying to find things that are new and exciting, and hopefully some of them will pop up.
Another technical question from me, Yanis. Could you comment on your experience comparing, say, high-C with high-chip and how you feel these techniques are useful or complementary?
Look, I mean, high-C gives you the ability to look at, you know, compartments and touch in a way that high-chip cannot, because high-chip can basically focus, as you know, on active sites when it comes to K27 or whatever antibody you have. You know, high-chip is basically tailored around your antibody, and it could be a cohesin, it could be a CDCF, it could be a histone modification, you name it. High-C will give you this more global view of things, including compartments within the nucleus. I prefer high-chip just because of sequencing depth and resolution. I mean, you don't have to kill yourself financially and time-wise to basically sequence billions of reads to get high-resolution maps, and you can do that cheaper and easier with high-chip, obviously. There are other ways to do that now. I mean, you've probably seen this technique that's called micro-C from Berkeley that actually, because of the change of the enzyme, that gives you the ability to look at very fine resolution, and it's something that we're starting playing with. But at this point, we're kind of going back and forth between high-C and high-chip, and between us, we're actually doing more high-chip than high-C as we speak.
And can you recapitulate your key findings that you've found from high-C by just using the high-chip?
Yes. And in much finer resolution, yes, obviously, yeah.
Okay. Well, I think we're at the end of questions, so we finished a little earlier than perhaps we had anticipated, but thank you for leaving so much time before the end of the hour, Yanis. It, again, was a really fascinating walk through this new era of leukemia genomics. So thank you again for participating, and we look forward to seeing you all again next week.
Thank you, Charles. Thank you, everybody, for being here. Thank you.